Hello, everyone, and welcome to Cascades Analytics Live. I'm Alexandre Stepanovich, founder of Cascades Analytics, a consultancy and media platform focused on the science and future of psychedelics and neuroplastogens. Here we interview the scientists, founders, and investors shaping the next wave of psychedelic-inspired therapeutics. Today, I'm very happy to welcome Professor Alex Kwan from Cornell University. For more than a decade, Alex's lab has been at the forefront of understanding how psychedelics reshape neural circuits in rodents using tools such as viral tracing, two-photon imaging, electrophysiology, and behavior. His lab's most recent paper in Cell makes a strong and important claim. Psychedelic-induced plasticity is not global or uniform. Instead, it is selective, strengthening some cortical networks while weakening others. It's a circuit-level view of plasticity that's far more nuanced than the idea of psychedelics as generic plasticity amplifiers. So let's dive in. Alex, thank you so much for being with us today. Is that okay if I call you Alex?
Yeah, please call me Alex, and glad to be here.
Yes, thank you. This time, everything seems to be working fine. So I wanted to, start broadly. Your recent paper, psilocybin triggers an activity-dependent rewiring of large-scale cortical networks, was published in Cell a few months ago. So congratulations. But what was the starting hypothesis you wanted to address when you began this study?
Yeah, the motivation of this study come from the series of work that my lab has done looking at how drugs like ketamine and also psilocybin and also 5-MeO-DMT, how they can change structural neuroplasticity in the brain. In the earlier study, we show that these drugs can invoke neuroplasticity. So we see the formation of dendritic spines, which is the putative site of the synapses where the neuron connect. But one of the big questions from those studies that we never addressed in the earlier work is where do these connections go? So synapses means that you have one neuron connect to another, but what are the identity of these neurons? We think that understanding what are the exact connections that's being strengthened and weakened and modified by psychedelic is quite important because ultimately we want to know what these drugs do in the brain. So that's sort of the impetus of this study. To figure out what are the specific circuits and pathway that's modified by these drugs.
And you think the plasticity in the cortex is different from plasticity, for example, in the striatum?
For sure, yeah. I think it's often. I think people get too excited about seeing things that can enhance neuroplasticity. But fail to acknowledge that our brain actually constantly change. Plasticity sort of engage in our everyday life. Many things can change your brain wiring. I would say many drugs do, not only psychedelic, but you have other drugs, even drugs of abuse like cocaine or heroin. Those might engage plasticity, more so maybe in the striatum, as you were suggesting, but also to some extent in the cortex as well. That's been shown in some really elegant historical work. Mm-hmm. But these kind of plasticity are obviously not beneficial, but instead maladaptive. It causes rewiring that lead to negative behavioral effects, leading to dependence. Mm-hmm. And then even more, I think even going beyond drug, I think, just again, us living our life, maybe listening to this live stream or taking a lesson or learning piano could rewire your brain. I mean, that's how we learn, and even far into adulthood, we have the capacity to learn and rewire our brain.
Mm-hmm. Great. Why don't we dive into the presentation you shared with me, and dive into this fascinating article. So, why don't you start? Yes.
So this is, I think, exactly the point that we were just making, that psilocybin is not unique, right, in terms of inducing plasticity. Mm-hmm. I think there's quite a lot of studies now demonstrating ketamine definitely does it. SSRIs, so other antidepressant like fluoxetine would do it. Drugs of abuse like cocaine. So these are classical work by Cope and Robinson back in the 70s and 80s, showing that they too change dendritic spines. And then finally, the act of learning. So I think it's very key for my lab to understand not only there's plasticity, because plasticity happens widely, but specifically what makes psychedelic special for psilocybin, at least, which is what we study here. Where is the plasticity happening? What are the cell type? What are the circuit that are being modified? So I think that is what makes the drug special and different from other things.
All right. Do you think all these molecules induce the same type of plasticity, or some strengthen some spines, whereas others rescue stress affected spines? Does different plasticity amplifiers, does that make sense?
I think that is a big unknown right now in the field. I think our understanding of drug-evoked plasticity is still in its infancy. Much of the work come in culture neurons, where we know the most, where if you add drugs to a dish, you can see, for example, spine growth or even dendritic outgrowth. But that's environment is obviously quite different from in the brain, and even similarities there is a bit unclear, right? In terms of in vivo study, we have done some work, but only limited to very specific brain region. I would say that this current work that we'll discuss that was just published is maybe even one of the first study, or maybe one of the first few study, let's say, that have used a more brain wire approach to look at plasticity in a kind of whole brain context due to a drug. So I think in terms of where that locus is at the circuit and cell type level, there's a lot unknown. Here we're just starting, and I think it will be interesting to compare eventually what happens to other manipulations like ketamine or fluoxetine, and how much is that it's similar or different. So here, I mean, again, trying to motivate the question here, we see that spine growth, which is, I just represent here with the magic wand. We see that plasticity and the induction of a new dendritic spine. But if you think about how neurons are connected, when you have a spine and a postsynaptic site, there must be something coming in from the axonal side, a presynaptic input, that form this functional connection. So the question is, what are these presynaptic inputs for the psychedelic-induced spines?
And do you think these are the 5-HT2A receptors you represented, or just any receptor?
Yeah, we drew a few things on there. You can see on the next to the magic wand, there are ion channels. So, and the spines, these are glutamatergic synapses. So they have AMPA receptor, NMDA receptor, and then you can see the squiggly line, which is my attempt at a GPCR. So that's supposed to be my representation of a 2A receptor. In a separate piece of work, we show that these 2A receptors are critical for the psychedelics effect on the structural plasticity. So I think they do play an essential role.
Let's move on to the next slide.
So this illustrates our approach for doing this. Previously, we do a lot of optical imaging to look at structural turnover. But in this case, we can't quite use that same method to look at the whole brain. It's a little too slow and too low throughput. Instead, what we do is we use a viral approach to map the connectivity in the brain. So what we do is leverage rabies virus. So these are engineered rabies virus. As many of you know, generally rabies virus is quite dangerous and quite toxic. But these ones, the toxicity has been removed and they're modified. But they maintain the ability, though, to jump synapses. So we inject the rabies virus into a set of cells in the frontal cortex of the mouse. And then what you can see here in the schematic is that the virus travel backward one synapse from the starter cell onto input cell. And then that transmission is through the connected neurons. So we know then and can trace and label these input cell with fluorescent protein. And we know then these input cells synapse onto the starter cell that we started with. The method is engineered, then developed maybe 20 years ago by Ed Callaway's and colleague in Salk Institute. It's quite widely used in neuroscience, but I haven't used quite often in pharmacology. So So I think here it just can be used to great effect to understand what a drug does.
Mm-hmm.
And I should mention, the people who actually did the work in the lab. So Kwan is the person who did the lab. He's wrote the work. Is a postdoc in the lab. And then we also collaborated with Allen Institute, who created the rabies virus construct and then produced these viruses. On the top, you can see the component of the virus. I won't go into too much detail, but we did screen 3 different types of rabies virus to find that this particular construct is really ideal for this purpose. Just labels very brightly and strongly. And then on the bottom, you can see the timeline of these tracing experiments. It involved injecting different kinds of virus and chemical in the beginning, but to prepare the animal, including the AAV helper virus and tamoxifen. But the key point is day 20 and day 21. You can see day 20, we inject either drug or saline. So the animal received 1 dose of psilocybin or 1 dose of vehicle. And then the very next day, we inject rabies virus to start tracing the connection. And then finally, 1 week later, we collect the brain. So what's key is the rabies only started tracing after the drug is induced. So any kind of connectivity that we trace with the virus is due to the drug and not pre-existing. And moreover, we give some time for the rabies to work. That's why we collect it 1 week after the rabies injection. To some people, I describe this tracing method as mapping the roads, like using Google car, Google self-driving car, Google Maps. I think it's a good analogy where, previously we were mapping like the roads, but one at a time in detail using optical imaging. Here, you can imagine the virus acts like self-driving car. They just go wherever they can. And then in that way, they map the circuitry for us in almost like an automatic way, but brain-wide manner. So that's why these viruses are very powerful, because they just go wherever the connection lies.
Mm-hmm.
And then, I don't know if you can play this, but this is a video actually, but it shows the some of the results of that injection.
Oh, okay.
If you can play it, or if you can just look at it, in white is the cells that are labeled, and then later on in red, you can see the starter cells. But I think even if you cannot play, it's okay. I mean, the video itself is included in a paper. I think you can get a sense on how the data look like when we inject these viruses into the brain, and then later on the imaging to tell where the virus has gone. It basically will show you a fly through of the brain and then where the label cells are.
Is there a way to know if the rabies virus is going to jump presynaptically uniformly? Since the neuron's architecture it's injected in it can be pretty complex, isn't the rabies going to choose maybe the less dense presynapses to go to, or the more dense? I don't know. Is there a preference for the rabies virus to choose his synapse to jump to?
That's an excellent question. So there is some detail about how the rabies virus work that we do not know in terms of just the biology of rabies. One of which is the point that you raised. One thing that is clear to us is that the rabies does not trace all connections, because if you look at a single cortical neuron in the frontal cortex, people know that it has about 10,000 or so synapses in one cell. So if you just do the math in terms of how many cells we start with and how many we end up tracing, you realize that we're not tracing all 10,000 connections. Then one could ask, so which of those 10,000s are you tracing? Are you tracing synapses that are stronger preferentially, or maybe you're tracing synapses that has certain proteins in them? So those are some aspects that we actually do not know. So, I mean, that is actually one limitation of the study. But I would argue that even given some of these limitations, rabies remain, I would say, the only way to do these kind of brain-wide tracing. So we should keep in mind there could be some preferences, but on the other hand, it gives us an unparalleled bird's eye view on what happens in the whole brain in terms of connectivity. But that is a good point.
Do you want to expand on this? Sorry, I didn't see whether a video
Yeah, this is another video. So I think, but if we can't see it, maybe I can describe a little bit of it and then we can move on.
Yeah.
But if you download the paper and the video is freely available, you can see green cell and then also red cell. Those that are red are these starting neurons in the frontal cortex. And then specifically the one with both red and green are the ones with rabies that are the starter cell. And then the one that's just green would be the one that synapse onto them. So those are the input cell. So if you can see the video, you see a lot more green cell because those are the presynaptic cell, input cell. And we're starting from about 1,000 cells in the frontal cortex, several thousand cells. So those would be the ones that are both red and green and just located in the front of the brain.
Mm-hmm.
One of the new ones in here, and I think several people already now told me the paper is complicated. I think that's also because the brain itself is a bit more complex, and we try to capture those nuances in these studies. Again, the point is, what are the cell type, what are the region? I'm not here just to tell you that plasticity has increased, but we want to find exactly where it is.
It's also a great step. I mean, to go from a network plasticity into a level below, which is the cell type plasticity. It's a great step. So it has to be complicated at one point.
Thank you. Yeah. But one has to recognize the type of cell that we previously showed that has the spine growth are not uniform, and there are several major subpopulations there. And two major subpopulations are the so-called PT and IT neurons shown here. Mm-hmm. The key distinction between these cell types is they send their output to different brain area. Mm-hmm. So the PT cell goes out of the frontal cortex, dive deep into the brain, go to places like thalamus, midbrain, pons. Some of them even go down to the spinal cord. So they're really a major output pathway of the brain. The IT neuron are quite different. You can see that their axon on the far right, that diagram, they kind of stay within the cortex and striatum, so they don't go outside. So these are the neuron that forms a lot of the recurrent loops that are within the cortex, because they also project within the cortex and stay within it. So both of these are types of pyramidal cell that we know exist, major subclasses. And we decided to do the rabies tracing starting from these major subclasses.
Mm-hmm. The spines are also different in both the spine density and the dendritic trees are different. The IT probably have more spines and more branched out dendritic tree than the PT, who have less spines. Is that accurate?
I would say maybe generally actually the opposite, but on average we're talking about. On the PT cell, what you can see, the PT cell, which is the far left in yellow, mm-hmm. You can see the dendrite, which is the thick tuft that comes up and then it fans out. Mm-hmm. It's a very beautiful dendritic architecture. We have a very elaborate dendritic tree. Mm-hmm. Whereas on the right in the blue, you can see the trunk, but sometimes it doesn't fan out so much. Mm-hmm. So the arborization is a little bit less elaborated. Mm. So, I mean, there's still spines up there, but it's just not as fully developed as a PT neuron. On average, that's true, but there is a lot of variability in these neurons.
Okay.
So this shows, again, some of the raw data. I have a few data slides before we talk about what the data mean. But just to show you how the tracing starts and where the starter cell lie. These are the on the left here is an image of red and green, right? The cell that are both red and green is the starter cell. Mm-hmm. So you can see that it's on the right side of the frontal cortex in the medial area. This corresponds to the anterior cingulate region of the mouse, which I think relate to the dorsal ACC in primates. And then you can also see now in this still picture, there are also some green cell around there, but also outside. These would be the presynaptic input cells. Mm-hmm. And what the quantification on the right show you is just that showing you that we made the injection correctly. We're starting from a pool of about 2,000 or so starter cell in these tracing. This is tracing from PT neuron using this FSF2 mouse. And then moreover, the injection, the starter cell is very localized. We're just starting from a particular brain region. You can see that most of them start from the medial area, the secondary motor cortex, the premotor cortex, or the anterior cingulate area, as I mentioned. Very few starter cell were found in other brain area. So yeah, this shows you just exactly where those starter cells are in the rabies tracing. And this is the kind of data we can get with the rabies tracing. Again, I won't try to overload with too much data, but just to give you a sense on how it looks like. Obviously, what we start with is we have these images with green dots and red dots all over the place. We use machine learning methods to count the number of dots in each of the locations. We segment the brain based on known mouse atlases, which give us 316 brain regions, each of them with telling us the number of green cell and red cell in each of the regions. And then we basically asked, how does that number of green cell change between the vehicle and psilocybin condition? If there are more green cell in the psilocybin condition, it means that those input have been strengthened relative to vehicle. If there are fewer green cell in the psilocybin condition, then that input pathway has been weakened. So here I show you that analysis for the PT neuron, the input that go to the frontal cortical PT neuron. What you can see is that some regions on the top, these are rank order now, show increase in the number of input cell, whereas on the bottom, some actually show decrease in the number of input cell. So some pathway get strengthened, some pathway get weakened. So this was actually a bit different from what we initially expect. When we started this study, we thought we would just have a few regions pop up, but it turns out that quite a few regions change. So this was one surprise where it seems like the plasticity is quite brain-wide and many regions are involved. And then the second surprise is once we start looking at what these regions actually are, you can see that it's also color-coded. And then the color is not random. It's because those regions that provide the input increase tend to cluster at certain networks, and those that decrease also tend to cluster in certain networks. So there's a very specific pattern on where the increase and decreases are in terms of the circuit level plasticity.
That's the first big finding here. You must have been relieved to see that the increase happens in for the same color code group or decrease.
Yeah, again, it was quite surprising because, as I mentioned, there are not many studies I know of that have used rabies to look at drug action. But there is one very prominent one by actually a co-author on this paper too, Kevin Baer. He and Rob Malenka has done a very similar study looking at cocaine. And in that study, they found that a few brain regions have very aberrant and strong changes in connectivity after a dose of cocaine. So this was why when we go in, we thought maybe psilocybin, we would also see just a few regions that get really large change in connectivity. And then we would go like, aha, like that's the pathway we should go and try to study. But it turns out to be a very, widespread network-specific effect that I think was, again, I don't think we have seen this type of change before, and I think it's quite unique to this drug, at least as far as we can tell. So, again, the rabies is quite rich in data. Each time we get about 100,000 green cell, the input cell. Wow. We did 8 animals for each condition. So there's a lot of cell, a lot of animals, a lot of brain region. Here, I just want to summarize the finding for all the rabies data. In the middle, what you see is the PT and IT cell, which is the main 2 subpopulation. I show you the input coming in and the change. So the different color is the different network. So you can see on the left, the purple one is the so-called medial network in the brain, which relates to the default mode network. Some people think it's the mouse homolog of the DMN. And then you have visual auditory area next. You have sensory motor. And what you see is those lines are pretty thick going into the PT cell, meaning that after psilocybin, those networks get strengthened. The next one is the lateral network, which is a homolog of a salience network involving the insular cortex, and then also the ventral medial prefrontal cortex. Those inputs tend to get weakened. You can see the line is fairly dim into the PT neuron. And then you can also see in the bottom, the PT neuron project to a range of other brain regions, again, deep into the brain output. On the right is the IT cell population. You can see that the input change is quite different. Some of the network now actually gets weakened, for example, into the IT cell, like the medial network, whereas lateral network actually gets strengthened into the IT cell. So the plasticity is not only network specific, but also cell type specific. And then finally, IT cell go back up into the other cortical area, because those are the ones that mediate the cortical communication.
Mm-hmm. And you're only looking at these two neuron populations. Why only these two?
I think these two populations, again, are two major subtypes. So they're not the only ones, but they constitute maybe about 50 to 60% of the pyramidal cell in the frontal cortex. So they're big groups. They're also non-overlapping groups, so which makes it good, because if they're PT, they're not IT. If they're IT, they're not PT. They have very distinct characteristic. And many other people have now shown that these cells likely responsible for different behavioral functions in the brain. They play different functional role. One of the main subtype we actually miss here that's excitatory cell type is the cortical thalamic neuron that go from the cortex to the thalamus. A lot of those are deeper in layer 6. And one of the interesting factors, actually, but those neurons don't have a lot of 2A receptors. They also turn out to be harder to target. So in this study, we did not study them, but it would be a useful place to look at in future studies. I should mention both, in this case, both PT and IT cell do express the serotonin 2A receptors. So both of these are really likely targeted as psychedelic.
And do the inputs on these two subtypes are the same?
The input, they receive input from similar network, but what you can see is that the effect of the drug is quite different on the two cell types. That's also, I should maybe explain some of the cartoon on the left and the right. So on the left for the PT, you can see the strengthening come from perceptual network, correlates of the DMN, the default mode network in the mouse. Weakening is the salience of the insula. And then all of these through the PT neuron go out of the brain. So how we interpret this change is that there is a stronger routing, a more direct routing of perceptual information, sensory information from the correlate of the default mode network that then directly translate into output action out of the brain. On the right, you can see for the IT cell, quite different, because you can see those same medial network now actually get weakened. But if you trace the whole loop, the IT output actually go mostly back into the medial network. So that whole loop from IT to medial network back to IT actually get weakened as a result of the drug. So we think there's also a weakening and in general just a lowering in the connectivity of these cortical loop involving the frontal cortex. We also was quite excited about this finding. I think so far, a lot of the discussion with psychedelic, I think, has been they increase plasticity, so they must be good for you. But I think here we actually have some evidence supporting the idea of weakening of the cortical loop, which I think might relate to some of the anecdotal discussion on how these drugs might reduce rumination and break cycle. And here we actually have some evidence anatomically that cortical loops are being weakened.
And if we extrapolate to human, here we're looking at the effects 24 hours after psilocybin exposure. So if we extrapolate, we could think that the antidepressant property of psilocybin is actually to remove you from rumination and expose you to facilitate action mode, I would say, rather than contemplative melancholic mode. Well, does that make sense?
Yeah, I would say the finding that we see here with the connectivity is consistent with some of those ideas. It gives some hint. I think to really draw it and link it to the behavioral change, we do need to do some more experiment that actually involve behavior, because right now we're just looking at connectivity and kind of synaptic structure.
Right.
So I would be a bit more conservative, but I think it is suggestive, and it highlights some specific pathway that one could go and study. With the cortical loop, I also want to mention that I think somebody pointed out on LinkedIn. I think it's good to also keep the nuance here that this is a particular loop that involves the medial network, the default mode network here. So there are also some other prominent loop and recurrent circuit in the brain, for example, sensory motor loop, that allow you to take in sensory information and then act with a motor action. So those are not the kind of loop that we study. So here we're talking about a very specific loop within the cortex that we find the weakening after the psilocybin administration in the mouse.
Right, and it's not taking into consideration interneurons, GABAergic populations either. So,
yeah, here we mostly trace the pyramidal cell, and I think the vast majority of the long-range connections within the brain across brain regions are mediated by excitatory cell in the cortex, at least. So this probably captures most of the long-range region-to-region connectivity, but no doubt some of the local connectivity would be scoped, and also the neuroactivity itself would be scoped by GABAergic cell types. And those are also targets of serotonin agonists because they have the receptors. So yeah, so that's sort of the rabies study where we look at the connectivity map change as a function of drug. And as I mentioned, there were a number of surprises. But one of the follow-up questions then is, we see this very intriguing pattern. How does that pattern arise? Like, why did we get this pattern and not some other pattern? I think understanding what makes the pattern give us some hint on, how the drug work. So one of the idea that we had was maybe it's the neuroactivity itself that changes the pattern. And the reason we suspect this is because we also have some other study where we look at brain-wide CFOS expression change after drug exposure. So CFOS is an immediate early gene where the expression would increase after neuron fire action potential. So if we correlate whether a region have increased CFOS after psilocybin versus if that region have increased number of rabies trace input cell, you can see a positive correlation that's statistically significant, suggesting maybe like if a region that's more active, then it tends to send more input to the PT neurons. So that was one of the things that we hypothesized, and we went on to test it further. So to test it, we did two things. This is one of the things we did. If it is indeed neuroactivity, one could go directly measure the neuroactivity and see if it has increased. So here we use a electrode called NeuroPixel, which is one of these fancier neuroelectrode, allow you to record hundreds of neurons at once. We put it in one of these input regions that we found have fairly large increase in connection strength with the PT cell, and we recorded those neurons that project. And indeed, we find that their activity is increased after the drug. I should note that this increase is acutely, right? So this is immediately after the drug was administered. We find that this connection, these neurons that projects from the retrosplenial cortex into the frontal cortex has increased. And then the other thing that we can show that the activity is important is it would predict that if we mess with the activity, then it would also change the rewiring. So here we use a chemogenetics method to shut down the activity in the retrosplenial cortex. So if activity is important for the rewiring, if we shut it down, we should also shut down the rewiring. And that's exactly what we found. So here on the top is a timeline of the experiment. You can see that we shut down the brain and then we give the mouse psilocybin, and then we do the same rabies tracing. What the bottom plot, the scatter plot show is that normally psilocybin increase the number of input from retrosplenial cortex, but if we shut down the activity in this region now, that rewiring is abolished. And then a neighboring region that we did not manipulate, the insula, the plasticity remains. So we can also, to some extent, shape the plasticity by changing brain activity of specific brain regions. Yeah, so this is sort of, I think, a good place to summarize the study. I think we show you some data and talk about some of the interpretations. In terms of the postsynaptic side, we didn't discuss, but we have papers showing the PT neurons are quite important. We talked a bit about the 5-HT2A receptors are quite important. But this study we just discussed is really about the presynaptic side on the input side. And the two main findings here, one is that the rewiring is very large scale, involve many brain regions, but it's very network specific. also cell type specific. So again, I think echoing your introduction, it's not just about the plasticity, but also where that plasticity happens. And then secondly, when we manipulate activity, we can change the drug-induced plasticity. I think that's a very interesting concept because it suggests that you can sculpt that plasticity. So when you give drug, it has certain effect. But if you pair the drug with neuromodulation, or maybe even some other kinds of intervention to change the ongoing brain activity, you can sculpt and change and steer that plasticity, perhaps for the benefit of using it as a therapeutic. So I think that idea here is demonstrated in an animal. And I know there's now a number of trials that's starting combining things like psilocybin with TMS. So I think there's a lot of room there to try to manipulate and steer the plasticity to improve therapeutics. So I think that's another major take-home message here.
Fascinating. You did some in situ hybridization with the 5-HT2A, the 1A, and the 2C, I believe. And you didn't see any strong correlation between expression of these receptors with the.
Yeah, you're correct. Yeah, so I show you one plot where we correlate the CFOS level with the rabies trace input level. We did a very similar plot to look at other things to see if they might relate. So we try to relate the amount of 5-HT2A receptor expression with the rabies input to see if maybe a region that had more 2A receptor would also then increase its input. And maybe some correlation with 2C. There's some significance there, but not for 1A and 2A. But in general, CFOS gave the stronger signal. I think what this means and the way to interpret this is not that these receptors are not important. Clearly, they're important because I think they're the main target for psychedelics, 1A, 2A, 2C. But it just tells you that I think it's the totality effect of these action on these receptors. So there's the polypharmacology, again, that we cannot ignore. Action on all of these receptors at different cell type ultimately shape the activity of the cell within that region, which then is the thing that correlate the most. So let's see if I can put it better. Basically, I think each receptor has some contribution, but the overall correlation is still strongest at the final output state, which is the firing rate change of the neurons.
Or could it be the 5-HT2A is located on other? The activated 5-HT2A are located on other nearby subtypes neurons, like the CT or maybe the interneurons, and
no, actually, I don't think that is the case because we actually now know quite a lot about where these receptors are located. So there are a large database from Allen Institute showing, and we know 2A receptors are located in both PT and IT cells.
Predominantly.
Quite strongly, but also in some other cell type like PD fast spiking neurons, NDNF interneurons, a few cell types, and also layer 6B neurons. So there are a few cell types that we know, and then also other cell types that does not express the receptors. So the projection neuron that we looked at that we think are quite important, driving the activity-dependent plasticity, they definitely have 2A receptor. But they also have other things, like, for example, the 1A receptor. So I think, the totality of that co-activation of these different receptors is what shaped the activity of those neurons.
So what is your next step now? What's the next design? What are you looking? Are you one day dreaming of understanding the whole all subtypes in the cortex?
I would say right now the thing that interests me the most is I would really want to extend the study to compare more drug classes and compounds. I think here we demonstrate for psilocybin it does a certain thing. But one other thing I'm constantly coming back to is even within psychedelics, there's a lot of nuances in terms of the behavioral effects. We have some study previously on 5-MeO-DMT, which is, again, another tryptamine classic psychedelic, but very different behavioral effect. It would be very interesting to see what that does in this context in terms of the connectivity change, whether we find some similarity and differences. My lab has also started some interest in some other substances with both short-acting and long-acting action, I think that are not traditionally considered a psychedelic. In the past, we look at, for example, ketamine, another one where there's acute behavioral effect and also long-term effect, but again, quite different type of acute effect. So I think looking at similarities and differences like this is quite important, because I think that it can give us some handle on what are the important principles that we can then use to develop new therapeutics.
All right. Well, thank you so much, Alex, for your time and your explanation. This is truly fascinating. I think it's a groundbreaking article into trying to, go another level from the network to the neurons. So, congratulations to you, and Happy New Year. And I hope I'll see you soon at a conference somewhere on the planet.
Great. Thank you.
Thank you. Thank you, everyone. Thanks for tuning in. If you have any questions, make sure to send me an email, and I'll be happy to review it. And next time we'll have Alan Nafayev from receptor.ai, the January 23rd. So make sure to mark your agenda. Thank you, and have a great weekend.